All right, this is the part 3, chapter 17. Just a quick short one on mutations. Different kinds of mutations are basically changes in genetic material, and point mutations are when you change just one base pair. So an A gets converted into a G, for example. And this is showing you how it leads to abnormal protein development. Here, if we have CTT, wild-type hemoglobin, that gets converted to the mRNA as GAA, makes glutamine. But if you convert the T or mutate the T into an A, then you get GUA, and that makes valine instead of glutamate instead. And as a result, that changes the amino acid sequence. Different types of point mutations can be base pair substitutions or insertions and deletions. And substitutions are less severe or harmful than our insertions or deletions because base pair substitutions are point mutations. And in fact, insertions and deletions are entire frame shift mutations. So it's not entirely accurate that this is found under point mutations on this slide. Here's a substitution like we had just seen. U instead of a C, for example, or A instead of a G shown there compared to the original wild type. Those are substitution mutations. Now, these can cause missense or nonsense mutations, which are kind of a larger grouping. Missense mutations are when the amino acid changes, but translation is continued. So that one amino acid gets changed. Instead of glycine, we have serine, for example. That's missense. A nonsense mutation is when a stop codon is mistakenly added in place of what should have just been a normal amino acid, which is where you see here. This would be a nonsense mutation. It creates a nonsense protein, where this creates a missense protein, if you like, one that's altered but still intact. Insertions and deletions, here you see an extra base gets inserted. That created a stop codon, in this case, nonsense. Here, this very extensive missense where you have every codon down the line from that insertion is altered and changed, and thus every amino acid down the line is now changed. The protein is very, very different. And here you see also an insertion or deletion, same way, of multiple nucleotides. These are frame shift mutations, and they are severe, very extensive damage. Mutations are spontaneous. It happened during DNA replication or repair, and they can be simple and harmful or not at all. Sometimes we have mutations that we call silent mutations, which don't cause any damage to genes. They're unaffected. Or even, for example, if you mutate the third base of a codon, so for example, CCA, if you took CCA and you made it into CCG, it codes for the same amino acid. That's called the wobble effect, that last base in the codon sequence. It still codes for the same amino acid, so the mutation didn't cause any harm. Mutagens are the things that cause the mutations, physical or chemical things in the environment or in the body that cause those mutations. So is it really one gene equals one protein or one enzyme? Not exactly. We learned a variety of reasons why, including the alternate hypothesis, alternate protein hypothesis. And here's a big summary of everything: transcription, translation, and so on. You can look at that on your own time. And we are done.